Comparing the human eye to a video camera fails to capture its true function. It doesn't simply record the world, it acts as a specialized biological engine, constantly processing raw physical data. Raw photons are constantly bouncing off objects in every possible direction. If this chaotic storm of radiation were to strike the optic nerve directly, the result would be blinding, incoherent white noise. To create an image from that noise, we rely on the cornea. This clear, dome-shaped tissue serves as a tough physical barrier, but it is also the eye's primary focusing tool. It performs roughly 70% of the light bending required for us to see. The cornea's rigid refraction provides the vital first step in our vision. It takes a chaotic storm of photons and transforms it into a controlled, usable beam. Immediately behind the cornea sits the anterior chamber. This space is filled with the aqueous humor, a clear, watery fluid. This fluid is part of a continuous cycle. It is produced by the ciliary body, flows through the pupil, and then exits through a drainage system called Schlemm's canal. This process maintains the intraocular pressure required to hold the eye's shape while nourishing the structures within it. The iris reacts to the intensity of light passing through this fluid. It uses a set of muscles to expand or contract the pupil, ensuring that exactly the right amount of light reaches the inner eye. This combination of fluid dynamics and a muscular aperture protects the sensitive inner eye from light overload, keeping the internal optical path clear and nourished. Directly behind the pupil is the lens. Its specific function is to fine-tune our focus for objects at different distances, a process known as the accommodation reflex. The lens is controlled by a circular ring of ciliary muscle. This muscle is connected to the lens capsule by a network of thin, elastic strands called zonular fibers. The Helmholtz relaxation theory states focusing on a near object causes the ciliary muscle to contract inward, loosening tension on zonular fibers, allowing the lens to thicken. Alternatively, the Shekar tension theory argues the muscle tightens equatorial fibers, pulling edges outward, forcing the center to bulge. This model of relaxation was first proposed by Hermann von Helmholtz. His 19th century biomechanical model remains the standard scientific explanation used today. This microscopic muscular tug-of-war provides an elegant solution to the physics of light, allowing for instantaneous shifts in our point of focus. Once the light is focused, it must cross the vitreous cavity. This massive compartment makes up 80% of the eye's total volume. The cavity is filled with vitreous humor, a gelatinous substance. Made of collagen and hyaluronic acid, it acts as a physical shock absorber, protecting the retina from rapid eye movements. While it provides essential structural support, this gel is not a static substance. It slowly breaks down and liquefies as a normal part of aging. However, the primary success of the vitreous is its transparency. It ensures the light survives the journey to the back of the eye without being scattered or lost. The light finally reaches the retina, striking two types of specialized photoreceptors: rods, which handle dim light and peripheral vision, and cones, which provide color and fine detail. Vision begins with a single chemical spark. A photon strikes a visual pigment, causing 11-cis-retinol to isomerize, physically straightening into a new configuration. This triggers massive biochemical amplification. One molecule activates hundreds of proteins, slamming shut cellular channels across the membrane. This process highlights a specific behavior of photoreceptors. While most neurons fire an electrical signal when they are stimulated, light causes these cells to hyperpolarize. They actually stop releasing their neurotransmitter. The system operates in reverse to what many might expect. The eye sees not by turning a signal on, but by turning its continuous dark current off. Retinal bipolar cells take this signal and split it into two parallel pathways: on pathways for light and off pathways for dark. This separation of contrast is the first computation required for sight. It provides the data the brain needs to detect edges and recognize the objects in front of us. To keep this process running, the retinal pigment epithelium works to continuously recycle spent retinal molecules, ensuring the eye is ready to process the next image. Millions of retinal ganglion cells collect these processed contrast signals. Their fibers merge to form the optic nerve, carrying the data to the brain. We never just passively look at the world. Every image is the result of a system that is constantly filtering, focusing, and translating the universe into electrical impulses.